So thank you very much, Adolfo, for your kind presentation, and thank you very much for Cybernet to accept my group here. It's a great pleasure to me, this first meeting, where I have the opportunity to meet with strong collaborators in many of our last project and present project. So I'm going to present here a short presentation because I am the last presenter. We are tired and we are on a rush. So firstly, I would like to present my group because in fact, as Dr. Lopez-Munain mentioned, we are a little bit different of many of the groups here. We are the translational medicinal and biological chemistry laboratory that now we are located and focus and work in the CIV in CSIC. And so our main focus and our main goal is to discover new and synthesis new drug candidates and pharmaceutical tools that allow us to cure or to understand one pathology. Also, recently we have started a new line of research in discovering and synthesize new biosensors that allow us to follow drug treatments or something, or protein movement or drug distribution along the organism. We are a multidisciplinary medicinal chemistry team with pharmaceutical, biologists, and chemists in our team, and we cooperate a lot with pharmaceutical companies. Also, as Dr. Lopez-Munain mentioned, we are very involved in technology transfer. Some of our patents were licensed since 2000 and create different spin-offs. And many of our compounds, or several of our compounds, reach clinical trials. And recently, 4 years ago, we have founded Anchor Pharma with a mission to done the preclinical regulatory task to start clinical trials for innovative molecules. So we work in different fields of research, but since '96, personally, I work in neurodegenerative disorders, in neurological disorders, and we have experience in developing new innovative drugs for different pathology like Alzheimer's disease, Parkinson's disease, different others. And recently, since 2009, in the last 10 years, we have an extensive program in amyotrophic lateral sclerosis. We work in a target-based way. That means that we are looking for compounds able to interact with different targets, and mainly our targets can classify the first one in different cholinesterase expression brain, and more recently in a specific cyclic AMP phosphodiesterases able to increase levels of cyclic AMP in the brain and also in peripheral tissues. And we have a great experience in protein kinases inhibitor. We start our programs in the late 19th in JSK3, but also we have different programs and different family of inhibitors for LRRK2, MLK3, TTVK1, and different others. The methodology that we follow in our team is the specific for a medicinal and biological chemistry team. So we identify the target selection, and after we design and synthesize newly compounds, we determine the biological screening, and we optimize the properties of this molecule to have an innovative drug that, if will be, in the best of the cases, the roots of the new drug that we will have in the available for patients. The target selection is based in scientific interest and also in the experimental accessibility that we have in our lab. And we have for discover and design the new compounds, we have different tools. So we have a potent chemical library with compounds that was synthesized since the last 30 years, specifically to target central nervous system. Also, we have potent programs to perform virtual screening, and we used to work with rational design, and we have different techniques of organic synthesis to produce the molecules. Also, for biological screening, we use not only different recombinant enzymes with non-radioactive methods, but now, and this was one of the reasons to move our lab to the CIV, we can use different cell culture, as we will see later. And also we have animal facility. We optimize the properties of our molecules. The first one is the potency, the biological potency, and only when the biological potency is somehow optimized, we optimize the synthetic method. And also we follow and we are very focused in determining drug-like molecules. That means improving all the ADME properties like absorption, distribution, metabolism, and excretion. And in that case, working in CNS, we pay attention to the potential of these molecules to cross the blood-brain barrier, and we have different screening methods to determine this property in our lab. With all these tools in our hands, we discover innovative drugs, and we work in different mechanisms of action, now in our own laboratory, but every time in a strong collaboration with many groups, national and international, and many of us are here in the room. So also we are very conscious of the importance of the patent, and we patent the molecules in the best point of the research project in order to be able to transfer to the pharmaceutical or biotech companies. So this is our general presentation of our group, and today I'm going to present to you the first results, short kind of results, that focus on our modulators of TDP-43. We have learned this afternoon how TDP-43, which is a nuclear protein and involved in many functions of transcriptional repression, is one of the main proteins that is not overexpressed, but with aberrant function in amyotrophic lateral sclerosis and other diseases, as we'll show you. So, in physiological condition, TDP-43, which is in a dynamic equilibrium between nucleus and cytoplasm, but mainly is located in the nucleus, participates in this transcription and transcriptional repression. But in pathophysiological condition, and we don't know why, in fact, this equilibrium is broken, and so TDP-43 migrates to the cytoplasm. Here there is the activity place of the cell and suffer different post-translational modification, like ubiquitination, like reduction of solubility, fragmentation, and hyperphosphorylation, and lack the ability to be in the nucleus. So our idea and our wide goal is to recover the homeostasis of TDP-43. And we are working not only in protein kinase inhibitor, but also in compounds able to modify the autophagy, mitophagy, agrophagy condition, for example, and also modulate cellular stress. So, some years ago, Manuel and Newman described the first aggregates of TDP-43 in motor neurons, but now we know that TDP-43 aggregates are not a hallmark of sporadic and familiar amyotrophic lateral sclerosis and frontotemporal dementia, but also is the main inclusion, is the main proteinopathy in Alexander disease, in Perry syndrome, in hippocampal sclerosis, in traumatic encephalopathy, and more recently, it's very implicated not only in Alzheimer's disease, but also in late, in this encephalopathy of aging patients. So, all this disease lack of treatment. So TDP-43, it's a good target for drug modulation. Okay, focus on protein kinases. There are different kinases that are reported to be involved in TDP-43 phosphorylation. Indirectly, we have JSK3, and in fact, our research starts by this point. But directly, casein kinase 1 is involved in the phosphorylation, and also cell cycle kinase 7, and more recently, tau tubulin kinase 1. For all these kinases, we have extensive programs of small molecule synthesis and discovery, and now we are present to you the result that we have by the moment in the casein kinase 1 program. To discover our first inhibitors, we implemented a reverse chemical genetic approach. And this approach is based, firstly of all, in implementing in our lab of an enzymatic method that was able to screen large number of compounds. In that case, we use recombinant human enzyme, and we select isoform delta and epsilon, which are the isoforms of these kinases more involved in neurological pathologies, and we implement in a luminescent assay development. And after, we screen our in-house chemical library, and we focus on chemical diversity of the different scaffolds. In that way, we will be able to discover the first prototype, which is this compound that I am showing you in the slide. And in this approach, in this reverse chemical genetic approach, the next step is to test if the molecules identified are able to reverse a phenotype in cell culture or in animal models. And in our case, we decide to show the phenotypic change in cell culture and having as an outcome the decrease on TDP-43 phosphorylation. In that way, we synthesize a large number of compounds. I am not to show you all the chemical syntheses that are at the back, which is a lot of work, but we have more than 100 new molecules with IC15 values in the low nanomolar and very selective. So we analyze our compounds, and we show that these compounds are ATP competitive, but they are very selective. We perform a wide screen panel of more than 450 different kinases with a very selective score index. So the first assays we show and we test in a cellular model using a neuroblastoma cell line and induce cell death with ethacrynic acid. That is an agent that is well documented that increases TDP-43 phosphorylation, and we treat it with our compounds that show a rescue from this neuronal death. And the rescue of the neuronal death was completely shown that was by decrease or simultaneously with decrease of TDP-43 phosphorylation. In that moment, as we have showed this afternoon, there are no well models where TDP-43 can be studied, and so the first model that we used was a Drosophile, and in collaboration with Dr. Lecourtois from Rennes University, we tested our compounds in a model that overexpressed human TDP-43 protein on brain, and so shorten is toxic, and so shorten the lifespan of the flies. And we test with different compounds, and we see how, or we saw how lifespan is increased, and this increase of lifespan is completely directed to the potency on inhibition of CK1, that means diminish of phosphorylation. So simultaneously, and entering the ALS field, the neuroinflammation and the role of microglia and astrogliosis, it's very important for the death of these motor neurons. And so in collaboration with Dr. Perez Castillo, here in the room, we tested in primary astrocyte and microglia primary cultures the decrease of inflammation activation in this primary culture. So we have a very new model to test our compounds, and this is based in human samples. This is developed in collaboration with Dr. Ángeles Martín Requero, that now we share this group here in Cybernet. And the fact is that we extract lymphoblast blood from patients and immortalize them, and so we can work with this kind of immortalized lymphoblast, and we have now a small biobank with more than 20 samples of sporadic ALS, and also more than 10 different familiar cell lines. And we have shown how the TDP-43 pathology is recapitulated in this model. And so, sorry. Wow, I don't know how to go back. Here, sorry. And so we can see how in the ALS samples there are an increase in TDP-43 phosphorylation, not only in the full length, but also in the truncate forms. Moreover, looking to the localization, we see how in the controls, TDP-43 is nuclear in the nucleus, but in the ALS samples is in the cytoplasm. So using this model, we tested the efficacy of our compounds, and the CK1 inhibitors are able to decrease in a dose-dependent mode the TDP-43 phosphorylation, but what is more important, recover the nuclear localization. That's recover the homeostasis of TDP-43 in this cell line. So, also in collaboration with the group of Dr. Eva Del Lago and Professor Javier Ruiz, also from Cybernet, we tested these compounds in transgenic mice that overexpress TDP-43 with this mutation under the prionic transgene, and so we tested the compound, and we observed, firstly, that the treatment of the compound ameliorates the general symptomatology, as the decrease in the weight is much less than in the transgenic animals, and also we see how motor neurons are preserved in the spinal cord, also using both NISL and also cholinacetyltransferase staining. And also we test how, and we show how this compound is able to decrease the inflammatory reactivity. Finally, we test that, in fact, TDP-43 protein phosphorylation is decreased in the transgenic mice after treatment, as you can see in this graph. So finally, to have a well drug candidate, you need not only to have a good, more or less, efficacy in animal model, but also good pharmacokinetic properties. And in that case, this compound is able to enter in the brain, both after oral administration or IP. So it is non-mutagenic. And so our candidate is ready or to further study. And in this moment, 2 days ago, we have received a cancer impulse in order to try to translate these compounds to the clinical environment. So, well, I would like to convince that TDP-43 is a good target, that some protein kinase inhibitors may have a key role in recover the homeostasis of TDP-43. Also, I try to show you that immortalized lymphoblasts is a good human-based model to study different drug discovery programs and mainly TDP-43 pathology, and that casein kinase 1 inhibitor, especially our compound, is a good candidate for further study. So this is my team. Thank you very much to the people involved here and also to the network that we have now in our community focused in develop ALS new therapies. And to all of you, thank you very much.
Thank you very much. I would like to say that this group is a different type of group in Cybernet, and it represents a magnificent and fantastic opportunity to collaborate in different fields for research group to develop research with a translational objective. Time for the last two questions. Any question? No question? One?
Thank you for the nice presentation. I was wondering if you have had a chance to test these compounds in ALS iPS cells and whether there is any effect on the neurons themselves. A second related question is that casein kinase inhibitors affect also non-canonical Wnt signaling, and I was wondering whether it may have also some impact on this pathway in other tissues, and what do you think it might be?
Okay, yes. In that moment, we are working with Sergio Gascon, and we are testing the compounds in motor neurons from human patients, but not obtained from iPS, but by direct reprogrammation from fibroblasts. So we will see what happens, and that's it. That's for the next meeting. And in the second way, by the moment, we have not seen any sign of toxicity in our compounds, but we have seen a good thing, we believe, and that is that casein kinase inhibitors sometimes may have neurogenic properties, and so may enhance the endogenous system of reparation.